Welcome back to the deep dive, everybody. Today we're going to be taking a look at, well, we can't really look at them, can we?
Not without a microscope.
Haha, right. We're talking about the world of microbes, how scientists study those tiny organisms out in the environment, doing their thing. We've got a whole chapter here laying out all the tools and techniques, and it is seriously fascinating.
Yeah, it's like a whole detective's toolkit for this invisible world, you know?
Yeah.
It used to be that we could only really study the microbes we could grow in the lab.
Right, the ones that would cooperate, so to speak.
Exactly. But this deep dive, we're going to see how researchers are tackling those that just won't grow on a petri dish. How do you study something you can't even see without serious magnification?
And these little guys are everywhere, right? They're running the show in so many ways, so understanding them, that's got to be crucial.
Absolutely. So this chapter, it lays out two main approaches. One is the classic, growing them in the lab, all nice and controlled. But the other, that's where it gets really interesting, studying them right where they live in their natural environment.
Okay, so let's start with those lab techniques. The first one they talk about is enrichment culture microbiology. Now, I'm no scientist, but when I hear enrichment, I think of like making something richer, more abundant.
And that's basically the idea. With enrichment cultures, you're creating a very specific set of conditions in the lab, tailoring the nutrients, the temperature, the gases, everything. You want to make it the perfect little vacation spot for the microbe you're interested in, while making it inhospitable for others.
So it's like you're setting out a welcome mat for only certain microbes.
Exactly. And if those microbes thrive, well, that tells you that organisms with those specific needs and abilities exist in your sample, in your environment. It proves they're there, even if you can't see them right away.
That's pretty clever. But then the chapter mentions this thing called enrichment bias. It sounds like maybe your welcome mat could accidentally attract the wrong crowd.
Yeah, that's a big issue. See, in this artificial lab setting, some microbes, they're just really good at growing fast. They might not even be the most important ones in the natural environment, but in the lab, they can take over, outcompete everyone else.
Like weeds in a garden, kind of.
Perfect analogy. So for a long time, our view of microbial communities was skewed, dominated by these fast growers, not necessarily the most ecologically relevant ones. So these new methods, they're trying to address that, give a more accurate picture.
So how do you stop those microbial weeds from taking over?
Well, one technique is dilution. You start with a very tiny amount of your original sample, and that reduces the chances of those fast growers just completely dominating everything. It gives the slower, maybe more specialized microbes a chance to get established.
So it's like planting fewer seeds so they all have room to grow, right?
Right. The chapter actually gives some cool examples of enrichment cultures. Bajerink's work with Azotobacter and the Winogradsky column.
Now, those names, they ring a bell from somewhere way back in my science classes.
Bajerink, he was studying nitrogen fixation, a really crucial process for life. He created a medium with no other nitrogen source except nitrogen gas from the air. So only microbes that could pull nitrogen from the air and convert it into a usable form, only they could grow.
Very selective.
Right. And the Winogradsky column, it's a self-contained ecosystem like a mini world in a glass tube. You get these layers, gradients of oxygen and other chemicals, and different microbial communities develop in each layer, depending on what they need.
So like a high-rise apartment building, each floor for a different type of microbe?
Exactly. It beautifully illustrates how these environmental gradients drive microbial diversity. Okay, so let's say you've enriched for the microbes you're interested in, at least the type. How do you actually isolate a pure culture, get just one kind of microbe by itself?
That sounds tricky.
It is, but we have these classical isolation techniques like streak plates and agar dilutions. The idea is to physically separate individual cells on a solid surface, like a petri dish with agar. Each single cell grows into a separate colony, and ideally each colony is from a single species.
So you're giving each little microbe its own personal space to multiply.
Precisely. Another technique is serial dilution in liquid media. You dilute the culture over and over until some tubes have, statistically, less than one cell per tube. Any growth there is likely from a single cell, a pure culture.
So like finding a needle in a haystack, but instead you're making the haystack smaller and smaller.
Exactly. And this dilution method is also behind the MPN technique, the most probable number, which uses statistics to estimate how many viable cells were in your original sample. But of course, once you think you have a pure culture, you got to be sure, right?
Yeah, how do you confirm that it's truly just one type of microbe?
Well, you examine those colonies under a microscope. Look at their shape, color, texture, make sure they're all the same. You do a gram stain, see if all the cells look the same, stain the same way. You even try growing them in different media, making sure they all behave the same way.
So it's a whole process of elimination, making sure nothing suspicious is lurking in there.
Right. And now increasingly we use molecular techniques like sequencing that 16S rRNA gene we talked about to get a definitive idea on that microbe. But all these techniques, they're still limited by what we can grow.
Right, those stubborn microbes that just refuse to thrive in a lab.
So that's where these more advanced single-cell isolation methods come in. And some of these, they sound like they're straight out of science fiction.
Yeah, the chapter mentions laser tweezers. I mean, seriously, laser tweezers.
I know, right? But it's a real thing. You use a focused laser beam to actually trap a single microbial cell, like a microscopic tractor beam.
Wow. So you can pluck out one individual microbe from the crowd?
And then you can move it, transfer it into a sterile environment, and try to grow it in isolation. Another technique is flow cytometry, which you might have heard of.
It rings a bell, vaguely.
It's used in a lot of fields. Basically, you pass cells, one by one, through a laser beam. You can then analyze each cell based on how it scatters the light or if it's fluorescent.
Okay, so like a microbial beauty pageant, judging them on their looks.
Sort of. But it's incredibly powerful because you can tag cells with specific fluorescent markers, like antibodies. So the flow cytometer can pick out and sort only the cells you're interested in. It's high-speed cell selection.
Very cool. And then there's microfluidic devices. That sounds miniaturized.
Extremely. Imagine tiny channels and chambers etched onto a chip. You introduce your microbes and they get trapped in these individual compartments. You can then watch how they grow and behave, even introduce different stimuli, all at the single cell level.
So you can see how one microbe reacts to changes in its environment all by itself? That's amazing.
It really lets you explore a microbe's fundamental niche, what it's capable of doing. And then there's high-throughput culturing, where robots do all the work, setting up and maintaining tons of different cultures at once.
Robots doing microbiology.
It's the future. It lets you try a huge range of conditions, way more than a human could manage, and that increases your chances of finding those rare or fussy microbes that need very specific conditions to grow. Okay, so we've covered those culture-dependent approaches, from enriching to isolating single cells. Now let's move on to the culture-independent methods. How do you study microbes without growing them at all?
That's what I'm really curious about.
One way is with general staining methods. The aim here is to visualize and count the total number of microbes in a sample, no matter what they are. It gives you a basic census of the microbial community, even if you don't know their names.
So it's like seeing a crowd of people but not knowing who anyone is.
Yeah, exactly. And they do this with fluorescent dyes. These dyes bind to nucleic acids, DNA and RNA, in the cells, and then they glow under a special microscope.
So you're basically making the microbes light up.
Right. DAPI is a common one, binds to DNA, makes the cells really stand out. SYBR, green eye, it's another dye, very bright, works on a wide range of microbes, even viruses.
Wow. And some of these dyes, they can even tell you if a cell is alive or dead, right?
Yeah, with viability staining.
Mm-hmm.
Use a combination of dyes, some that can only enter damaged cells and some that stain all cells. By looking at the different colors, you can see which ones are alive and kicking and which ones are, well, not.
So it's like a microbial health check, seeing who's thriving and who's not so much. But what if you want to go beyond just counting and actually identify specific types of microbes?
That's where foliage comes in. Fluorescence in situ hybridization.
Okay, that one sounds familiar too, but I need a refresher.
It's super cool. You use these fluorescently labeled DNA probes. They're like little homing beacons that target specific RNA sequences inside the cells, the ribosomal RNA or rRNA.
So each probe is designed to find and stick to the RNA of a specific type of microbe.
Right. And these RNA sequences, they're like fingerprints, unique to different groups of microbes. By carefully choosing which part of the RNA to target, you can make probes that are very specific, identifying individual species or even broader groups, like families or whole phyla.
Wow. So you can actually see which microbes are which right there in the sample.
And the brightness of the signal can even tell you something about how active those cells are, because growing cells have more ribosomes, more RNA to bind to. But there are even more advanced versions of fish. There's cardfish, which amplifies the signal so you can see even those microbes that aren't very active or abundant. And then there's bond catfish, which lets you see which microbes are actively making proteins.
So you can actually see who's busy working and who's slacking off.
Exactly. And then there's Klasskai fish, which uses combinations of probes and special imaging techniques to identify hundreds of different species all at once. It's like taking a microbial group photo.
Amazing. Okay, so we've seen how you can directly visualize microbes under a microscope. But what about those culture-independent molecular analyses, the ones that rely on DNA and stuff?
Right. So one of the most powerful tools here is PCR, polymerase chain reaction. You've probably heard of it.
Yeah, it's famous, but I'm fuzzy on the details.
Well, in microbial ecology, we use PCR to amplify specific genes from all the microbes in a sample. We often target those RNA genes again because they have regions that are the same across all microbes and regions that are unique to specific types.
So you're basically making millions of copies of these fingerprint genes from all the different microbes in your sample.
Exactly. And then we have to analyze all those copies to figure out who's there. Older methods like DGGE, TRFLP, and ERISA, they were like trying to sort those gene copies by their subtle differences, even without knowing their exact sequence.
Okay, like sorting Lego bricks by size and shape, even if you don't know what the final model is.
Great analogy. Another way was to create clone libraries, where each individual gene copy was put into a bacterium and then sequenced.
Well,
But that was really laborious.
talk about tedious.
Right. But then came next generation sequencing, and that was a game changer. These new methods can sequence millions, even billions of DNA fragments at once, giving us a much deeper and more complete picture of the microbial community. They've even revealed the rare biosphere, all those microbes that are present in tiny amounts but might still be important.
So like finding those rare hidden pieces in a massive jigsaw puzzle.
Exactly. And then there's quantitative PCR or qTCR. It not only detects the presence of specific microbes, but also tells you how abundant they are in the sample. So it's like a microbial census, telling you not just who's there, but also how many of them there are.
Very cool. So those molecular methods, they've really blown open our understanding of microbial diversity, haven't they?
They absolutely have. And then there are microarrays, which are like miniaturized labs on a chip. You have thousands of tiny spots, each with a specific DNA probe, and you can see which microbe's DNA binds to which probes.
So it's like a microbial dating service, seeing who matches up with whom.
Uh-huh. I like that. There's the phylochip, which targets RNA genes to look at evolutionary relationships. And then there's the geochip, which targets genes involved in important metabolic processes. It can tell you what kinds of functions the microbial community might be capable of.
So it's like a skill assessment, seeing what talents the microbes possess.
Exactly. But it's important to remember that just because a gene is there doesn't mean it's being used. It's like having a toolbox. You might have a hammer, but that doesn't mean you're building a house right now. And that brings us to environmental multi-omics, which tries to get a more complete picture by combining different types of data.
Okay, this sounds like a really powerful approach.
It is. We've talked about metagenomics, which sequences all the DNA to identify all the genes present. But then there's metatranscriptomics, which looks at the RNA to see which genes are actually being expressed, being used to make proteins.
So metagenomics tells you what the microbes could be doing, and metatranscriptomics tells you what they're actually doing at that moment.
Exactly. And then there's metaproteomics, which identifies and quantifies all the proteins present. These proteins are the real workhorses of the cell, carrying out all the important metabolic functions.
So it's like seeing who's actually out there on the factory floor doing the work.
Precisely. And finally, there's metabolomics, which analyzes all the small molecules, the metabolites, that are produced and consumed by the microbes. It tells you what's being made and used, giving you a glimpse into the metabolic pathways that are active.
Wow. So it's like taking inventory of the raw materials and finished products in a microbial factory.
You got it. By combining all this omics data, you get a much more holistic and dynamic view of the microbial community. You see who's there, what they're capable of, what they're actually doing, and how they're interacting with each other in their environment.
Okay, so we've covered how to identify the microbes and figure out their potential. But the last part of the chapter, it focuses on actually measuring what they're doing out in the environment, their activities, in real time. The first section here is about bulk activity measurements. So what are we trying to learn here?
The goal is to get an overall sense of the metabolic rates and processes that are happening in an entire microbial community. It's like measuring the total output of a factory, rather than focusing on individual workers.
Makes sense. And the chapter mentions using isotopes and inhibitors for these kinds of measurements. Can you explain how that works?
Sure. One common approach is to use radioactive isotopes. You introduce a substrate, like a food source, that's labeled with a radioactive atom, and then you track how that radioactivity gets incorporated into different metabolic products over time. For example, if you want to measure sulfate reduction, you can add sulfate labeled with radioactive sulfur and then measure how much radioactive sulfide is produced.
So it's like giving the microbes a special meal that glows in the dark and then seeing where that glow ends up.
That's a great way to put it.
Right.
And stable isotopes, which aren't radioactive but are slightly heavier than normal atoms, can also be used. You can track their incorporation into different molecules using a mass spectrometer. Another approach is to use chemical inhibitors. These are molecules that block specific enzymes or metabolic pathways.
So it's like throwing a wrench into the machinery to see which part stops working.
Exactly. By comparing the activity in a sample with the inhibitor to a control sample without it, you can figure out how important that particular pathway is in that environment. And of course, it's crucial to have proper controls, like samples with killed cells, to make sure you're actually measuring biological activity and not just some random chemical reactions.
Okay, so that gives you a big picture view of the community's metabolism. But what if you want to zoom in and see what's happening at a much smaller scale?
That's where microsensors and nanosensors come in. They let us measure things like oxygen, pH, or nutrient concentrations at a very fine resolution, even down to the level of individual microbial cells.
Wow, so you can actually see how conditions vary from one spot to another, even within a tiny microbial community.
Right. Microsensors are like tiny probes that you can insert into things like biofilms or sediments. They're usually electrochemical, meaning they measure electrical currents that change depending on the concentration of the molecule you're interested in.
So you can create a map of the chemical environment, seeing where the oxygen is high, where the pH is low, and so on.
Exactly. And nanosensors are even smaller, often based on fluorescent nanoparticles or other nanoscale materials. They can be incredibly sensitive, allowing you to measure things like oxygen levels right at the surface of a cell. The chapter even mentions an assay for measuring nitrogen fixation using acetylene gas, which the enzyme nitrogenase can convert into ethylene, a gas that's easy to measure.
So you're essentially tricking the nitrogen-fixing microbes into revealing themselves by giving them this special gas to work with. That's really clever.
It is. And then there's stable isotope probing, or SIP, which is a really powerful technique for linking specific microbes to specific functions. You see the microbes a substrate that's labeled with a heavy isotope, like carbon-13 or nitrogen-15.
So it's like giving them a meal that's slightly heavier than usual.
Right. And the microbes that use that substrate will incorporate the heavy isotope into their DNA, making their DNA denser. You can then separate the heavier DNA from the lighter DNA using centrifugation.
So the microbes that ate the special meal get separated from those that didn't.
Exactly. And then you can analyze that heavier DNA to see which microbes took up the labeled substrate, which means they're the ones carrying out that specific metabolic process. You can even combine SIP with other techniques, like metagenomics, to learn more about the genes and pathways involved.
So you can not only identify who's doing what, but also how they're doing it. That's incredible.
It really is. And then there's the really cutting-edge stuff like nanosims and fish sims, which can measure the elemental and isotopic composition of individual cells with incredible precision. You can actually see which atoms are present in each cell and where they came from.
Wow, that's mind-blowing.
I know, right? It's like having a microscope that can see individual atoms. When you combine nanosims with fish, you can first identify a specific cell based on its rRNA sequence. And then you can analyze its isotopic composition with nanosims. It's like a double whammy, giving you both the identity and the activity of a single microbe. The chapter also talks about other techniques, like Raman microspectroscopy, which can tell you about the biochemical composition of a cell, and MARFISH, which uses radioactive tracers to see which cells are taking up specific substrates. Flow cytometry can also be used to analyze and sort single cells based on things like FISH signals, and single-cell genomics can give you the entire genome of a single microbe, even if you can't grow it in the lab.
Wow. This has been an incredible journey through the world of microbial ecology. It's amazing to see how far we've come in our ability to study these tiny but incredibly important organisms.
I agree. This chapter really highlights the incredible diversity and ingenuity of the techniques that microbial ecologists are using to unravel the secrets of the microbial world. And it's not just about satisfying our curiosity. This knowledge has huge implications for fields like medicine, agriculture, and environmental science.
Absolutely. Understanding microbes is essential for tackling so many global challenges, from disease to climate change, and who knows what amazing discoveries are still waiting to be made.
Exactly. The more we learn about microbes, the more we realize how much we don't know. But with these powerful tools, we're getting closer to understanding their intricate roles in our world and the potential they hold for solving some of our biggest problems.
That's a great point to end on. Thanks for joining us on this deep dive into the fascinating world of microbial ecology. I don't know about you, but I'm feeling inspired to go out and explore the microbial world a bit myself.